Bismillah ar-Rahman ar-Rahim. So this is Fatima Khdar again, and today I'm going to talk to you about molecular techniques. Okay, so let's now go to PCR, polymerase chain reaction. So in PCR we got this DNA fragment, and we want to amplify it into large number of copies. We want to copy it, okay? So we first start with separating the strands. So put it in a temperature of 95, which will cause the two strands to separate. Then we put it in a temperature of 55, which will cause the primer to anneal with the strand. Then you put it in a temperature of 72, which will cause DNA polymerase to elongate this strand. So you got this double-stranded DNA. So from one copy, we double the number, we keep repeating the cycle, doubling the number until we amplify it into a large number of copies. Moving to plotting, starting with Southern blotting. Okay, so in Southern blotting, what you want to see whether a particular sequence of DNA exists or not. Let's say whether gene A exists or not. Okay, so how will we do this? So let's say we got this DNA that is very tall. We go and we use an endonuclease. We will cut it into fragments and we put it in gel electrophoresis. Gel electrophoresis is basically a plate where DNA fragments will be separated according to their charge and size. Then we again put it in a filter. The idea of the filter is just to better visualize these fragments. Okay, so now what will we do? Concentrate. We got this complement of gene A. Okay, we radio label it and we put it in this filter. If gene A exists in this patient, the complement will bind and the filter will shine up, telling you gene A exists in this patient. Okay, so again, DNA is cleaved, put it in a gel electrophoresis, then in a filter, then a probe is used and radio-labeled to see whether gene A or the sequence exists or not, and how does it look like, tall or short, as you saw in your slides. Northern blotting is the same idea, but the difference is that you are looking for an mRNA sequence. Does an mRNA sequence exist in this patient or not? Moving to Western blotting, you are looking whether a particular protein exists or not. Okay, and the way to do it is that you put the proteins here, and you got this antibody against this specific protein. You put it here, and if this protein exists, it will bind to it and it will shine up again. And this is the mnemonic snowdrop that you already got in your biochemistry. Moving to ELISA. In ELISA, I want to see whether an antigen or an antibody exists or not. So let's say this is the plate, or this is the well, let's say, and this is the antigen. So this patient is having this antigen. Okay, we add the antibody, which is attached to an enzyme, and we add a substrate. Now, if this antibody is attached to this, if this antigen exists, the antibody will attach to it, and the enzyme will be converting the substrate into another substrate, which are yellow, and the well will look yellow. So if you are seeing the well yellow, this means that the patient is having the antigen, because the enzyme is working, because the antibody bound the antigen. Finally, we are in CRISPR. CRISPR is a very amazing technique where you got to remove a mutated sequence and put the right sequence instead. Unfortunately, this is limited to the lab. Hopefully, there are many researchers that they are trying to cure genetic diseases by applying this in the clinical field. I really hope them all the best. I really hope you all the best in this in molecular medicine. And please, if you have any questions, email me, and I really hope you got something from this video. And good luck!